So my name is Ernest Smith. I'm an alumnus, and I'm the current president of the School of Medicine and Dentistry Alumni Council. It gives me great pleasure to introduce Dr. Jill Weimer today as our alumnus guest speaker. Dr. Weimer is nationally recognized neuroscientist and biotechnology leader, advancing treatments for rare pediatric neurological diseases. She is currently the Chief Scientific Officer at Amicus Therapeutics and also Senior Director of Therapeutic Development at Sanford Research, where she oversees preclinical research and translational development programs aimed at conditions such as Batten disease and neurofibromatosis type 1. She will be presenting today From Rare to Remarkable, Turning Research into Therapies. We will have time for Q&A at the end, and we'll come to you with handheld microphones if you have questions.
Thank you, Ernest, for the introduction, and thanks to the advancement team in particular for inviting me here today to tell you about some of the exciting work that actually started when I was a graduate student here at the University of Rochester and continues amongst a number of folks that are in the audience to continue to pave this way. So as Ernest mentioned, today I'm going to tell you about work that I've been doing on therapeutic development as a basic neuroscientist, and for anybody in the room that is an early discovery scientist. We always have this utopian dream that we'll be able to see things from our bench advance into the clinic. For many of us, that fortunately never happens. We pass that baton on to others and our advancements move forward. But today I'll tell you about 3 programs that we've actually been able to advance into the clinic for Batten disease and sort of that remarkable journey. I actually, in addition to being an alumnus from the School of Medicine and Dentistry, I actually started out on River campus. I came here in 1993 thinking that I was going to be a mechanical engineer. Within a year, I quickly discovered that I hated math. And as an engineer, it's something you need to be skilled at. My family quickly asked, coming from rural farm country, Missouri, why did you move all the way across the country if you're not getting a mechie degree? And I looked around, and at the time, the University of Rochester was one of 2 schools in the country that had undergraduate degrees in neuroscience. I love basic biology, and I said, well, I'm going to become a neuroscientist. And they said, what are you going to do with that? And I said, for about a week, I said I would go to medical school. I quickly discovered, though, that my passion and drive are actually at the bench in early discovery work and understanding how the brain develops. So after I finished my undergraduate, I wasn't completely clear on what I wanted to do. So I, as an undergraduate, had been fortunate to work with Paul Coleman, who at the time was in the Department of Neurobiology and Anatomy, and Carrie O'Banion. And so I sent an email over to them and said to Paul saying, I'm looking for a position, and he welcomed me into his lab. I worked on Alzheimer's disease for 3 years. We were doing microarrays before Affymetrix existed. So we would go to the morgue, collect autopsy samples from Alzheimer's patients, isolate single neurons, do amplification of their genome, and put those onto custom-made microarrays to look for gene changes in Alzheimer's disease. That really fueled my passion for neuroscience even more. I realized that I wanted to be at the bench developing those questions, so matriculated into the PhD program in 2000, just as this wonderful building that we're standing in was open. And I moved into the 2nd floor here under the leadership of Howard Federoff, who was a gene therapy expert, and a new, brand new faculty member who was recruited into that center by the name of David Pierce. David actually trained with Fred Sherman here at the University of Rochester in a yeast genetics lab and had uncovered that there was a yeast homologue for one of the common Batten disease genes, CLN3. He had never touched a mouse in his life. So I came to his lab to actually characterize the first mouse models for CLN3 Batten disease. And you'll see those peppered through this talk. That torch has been carried here at the University of Rochester by John Fox and Ed Freeman's team, who are actually now studying that mouse model and human models for looking at EEG and auditory changes in CLN3 Batten disease. After I departed U of R, I wanted to delve more into developmental neuroscience. So I went to the University of North Carolina, where I did my postdoctoral fellowship on mechanisms mediating brain development and migration of newborn cells in the brain, neural stem cells in the brain. Right as I was finishing up my time there at UNC, my advisor from the University of Rochester, David Pierce, was recruited to, of all places, Sioux Falls, South Dakota, to start a research center. He called me up one day and said, we work really well together. Would you come and help me launch the center? I said, you're from England. Do you know where South Dakota is? I don't know if they have research there. So I committed to going there for 2 years to help him launch a research program. As of next month, I'll have been there 16 years. It sucked me in and has been a really great place to launch my research career and really build up the science that I'm going to tell you about today. But as Ernest mentioned, and you'll see this throughout the talk, a number of the programs that we have developed have made it into clinical trials. And in 2019, several of the gene therapy programs that we developed were acquired for temporarily by a Philly-based pharmaceutical company. So I also serve as the Chief Science Officer of Amicus Therapeutics, my lab space based in Philadelphia. So none of the work that I'm going to talk about today is related to Amicus. It's really related to the work that I've done in Sioux Falls. So while I was here at the University of Rochester, I got introduced to Batten disease. And so just stepping back one step, this is a rare neurological disease. For those of you who don't work or are familiar with rare diseases, these in the United States, the definition of a rare disease is any disease that afflicts less than 200,000 individuals. And surprisingly, there are over 7,000 rare diseases worldwide. So in the US, rare diseases impact 1 in 10 individuals. About 2/3 of these are pediatric, and there are, in most cases, no known curative treatments for these diseases. Batten disease is actually a family of neurological diseases. Collectively, they're actually the most common pediatric neurological disease. And imagine your child in their 4 to 5 year old, seemingly normal, hitting all of their milestones, and they develop a pediatric form of Alzheimer's disease. They start to regress. So maybe they're walking. Today, we're going to talk about CLN3. Those kids start to go blind very early, around the age of 5 or 6. They have sleep disorders. They develop behavioral issues, severe seizures. They become wheelchair bound in their early teens, and they usually do not survive past their mid-20s. And so it's just a horribly devastating diseases. There are 13 different forms of Batten disease, each caused by mutations in different genes. These are lysosomal storage diseases. So thinking back to your biology classes, the lysosome is our recycling center in our cells. So the genes that are involved in Batten disease impact either directly lysosomal enzymes, like lysosomal structure and trafficking, or some of the biology. So ultimately what ends up happening is cells throughout the body actually accumulate junk, garbage in their lysosomes, which impacts their function, and ultimately those neurons die. This is almost entirely a neurological disorder. So one of the things that I won't have time to talk about in our lab, we've spent a considerable amount of time trying to understand why neurons are more impacted than other cells in the body in Batten disease. And we think that these Batten disease genes serve alternative functions in neurons, helping to traffic cargo like synaptic vesicles and lysosomes to the nerve terminal, which have to be able to travel over long distances. So my lab started out, as I mentioned, really as sort of a basic neuroscience lab, really doing a lot of that initial work that I mentioned, looking at these unique functions of 3 of the CLN proteins called CLN3, CLN6, and CLN8. And we've used tools like BioID to be able to dissect the movement of these proteins in neurons as they traffic up and down axons. We've then taken that biology to start to jump into therapy development. And I like to bend therapy development into 2 categories. Therapies that are actually treating the symptoms of the diseases, things like small molecules that might help with neuroinflammation or neurodegeneration, and then genetic medicines that we'll talk about today that really get to the root cause of these diseases. And so we've been fortunate to collaborate with a number of phenomenal individuals on genetic therapies that I'll focus on today, but much of our work is actually pivoted to small molecules. Once we were able to get a number of therapies into the clinic, I started to realize that as a basic scientist, my job does not stop when I pass that baton to the clinical team. There are a lot of things that we as basic scientists can be doing that actually help drive and accelerate these clinical trials. This includes making better animal models. So our team has made a number of mouse models. Today, I'll talk about some of the pig models that we've developed for Batten disease that we're now using to do biomarker discovery work to be able to identify blood signatures that can be used in the clinic to help accelerate clinical trials. So unfortunately, I won't have time to talk about all of this or in much depth, but feel free to ask questions at the end if there's things that I don't touch on that you're interested in, or grab me afterwards, and I'm more than happy. We're scientists, we love to talk about what we do. So I'm gonna start off talking about some of our initial bench work in genetic medicines. So I mentioned today that there are a number of Batten disease proteins. Some of them reside within the lysosomes, where you would think that they were most significantly impacting and leading to lysosomal disease. But some of them, interestingly, don't even reside in the lysosome. For instance, CLN6 and CLN8 that I mentioned, my lab does a lot of work on, are resident to the ER. And we think that they're important for actually processing and handing off some of the lysosomal enzymes in their proform to the lysosome. If you lose those key lysosomal enzymes are not actually trafficked properly to the lysosome. CLN3 is the one that I'm going to talk about today. We are just now, I started on this in 2000 with a mission of when I started graduate school at trying to understand the function of CLN3. And 25 years later, I think we're just finally starting to open that door and get a peek at what this protein does. So imagine trying to cure a disease where you don't even know what the primary function is of the protein that you're working with. So Batten disease, I mentioned, a lot of times the analogy we use is that it's like a pediatric form of Alzheimer's disease. So for the neuroscientists in the room, when we think about Alzheimer's or Parkinson's disease, we often think of it as a disease of the synapse. And so you have some sort of primary insult that impacts the connection between our neurons. And neurons are extremely hardy. It takes them years to die. And so you get this insult at the synapse, that sends up a red flag, which then recruits our glia cells. So our microglia are kind of like the first responders when that synaptic insult occurs, and neural inflammation starts to ensue. Astrocytes that are shown here in green kind of sit with their little fingers poked into those synapses. They start to respond as well. So in Batten disease, the first changes that you actually see are microglial activation, shown here, and astrocytic activation after you see sort of that lysosomal accumulation. We use one of the constituents of that storage material is called mitochondrial ATP synthase. So you'll see throughout, I'll abbreviate that sub-C to indicate that lysosomal storage material. So we use these as key markers of the disease. So I mentioned to you one of the places that we've really led the charge is in using viruses to introduce or reintroduce that faulty gene. So in this case, what we've done is create a gene therapy, an adeno-associated virus that targets specifically neurons, and we've reintroduced the human CLN3 gene. So we freehand deliver this into the brain of, into the lateral ventricles of mice day after they're born, and then we track them over time out to normal mice live to about 2 years. These CLN3 mouse models that many of us work with, unfortunately, do not fully recapitulate the disease. They have a lot of the pathological markers, but they don't have a lot of the behavioral markers. And that's where work that Ed Freeman and John Fox's team are doing here at the U of R to look at EEGs and brain activity are actually showing us that these mice are probably more similar than we think to a CLN3 patient. So I can tell you quickly that we went through and reintroduced the human CLN3 into the brain of these animals, looked at some of those secondary or tertiary cellular defects like storage material. And you can see 4 months after delivery or 8 months after the delivery of that gene therapy in 2 different brain regions in the somatosensory barrel cortex of the mouse or the thalamus, the VPL nucleus of the thalamus, that you can get almost a complete prevention of that storage material accumulated after delivery of that AAV9 CLN3. Same thing for microglia. Interestingly, the microglia only gives about a half rescue. One of the reasons that we think this is happening is that the AAV9, I said, is really good at transducing neurons and astrocytes, but it does not transduce microglia. So the gene therapy is actually not even getting into these cells, but by making the neurons healthy, maintaining those synaptic connections, it's almost like rising tides lift all boats. You can actually see an improvement even in the microglial activation to where it's still dampened down. But this is where I mentioned we're working on some small molecules because we think that gene therapy in and of itself is not going to be the complete cure. You're still going to have things that tackle things like neurodegeneration and neuroinflammation to kind of augment the gene therapy. Astrocytic activation shown here. You can see that this too is completely dampened down at 4 and 8 months of age in these animals. So we had a great gene therapy, then in partnership with Nationwide Children's Hospital at the time, and later Amicus Therapeutics, we were able to file for an IND and move this into a clinical trial. So this clinical trial is actually still ongoing. I'll talk about some of the results from the clinical trial next, but just to sum up sort of the bench side of things, we've been able to advance gene therapies for both CLN3 and CLN6 Batten disease. We're actually IND ready for a third gene therapy for CLN8 Batten disease, and really been able to develop this diverse pipeline focused on CLN1 through CLN8. I'll mention in the clinical piece a little bit of the work that we've been doing in collaboration with Michelle Hastings on exon skipping antisense oligonucleotides, similar to some of the work that John Lewick in the audience is doing here for cystic fibrosis. We've also started a very robust collaboration with David Liu's lab at Harvard, who is the inventor of base and prime editing. And so we're using gene editing tools to go back in and with precision medicine correct the mutations in our CLN2 mouse and pig model. I mentioned that our lab is also doing a lot of work on small molecule development. Having a foot in pharma now has helped me realize that medicines for rare diseases are almost impossible to get across the finish line. The amount of investment that goes into drug development and clinical trials are a losing battle for pharmaceutical companies. So the approach that my team took several years ago was to say, can we identify small molecules that actually could treat all forms of Batten disease? Rather than just focusing on one, maybe that's ultra rare and there's maybe 25 patients in the United States, a drug company is never going to get that across the finish line. So what we've done is identified a number of small molecules that actually show benefit for all forms of Batten disease. But even more exciting, we've started screening these in other lysosomal storage diseases like Pompe, Fabry, Gaucher, and they also show therapeutic benefit in those diseases. We thought, hey, let's step even further outside of the rare diseases and look and see whether these could have therapeutic benefit in diseases that just have lysosomal dysfunction, like Parkinson's disease or Alzheimer's disease. And when we screen diseases like Parkinson's models, we're also seeing therapeutic benefit for those. So stay tuned for those. We actually hope to be able to spin these out into a small company in the near future and advance these towards a basket trial for Batten disease first, and then some of these other lysosomal diseases. So I mentioned to you that the next exciting step that we got to take was to move these medicines from the bench to the bedside and into the clinic. So in 2015, after a short 8-month preclinical period, probably the fastest gene therapy program in the history of gene therapy, we were able to move to an IND for CLN6 Batten disease. That program has enrolled 12 children, and we're actually partnered with Alcyone Therapeutics and the Gray Foundation to continue to advance our CLN3 and CLN6 Batten disease programs further. Just staying on the theme of CLN3 Batten disease, this program has enrolled 4 patients. And this kind of shows you that natural history progression that I mentioned to you, where the children start with visual loss and behavioral problems, progress to seizures, anxiety, and motor movement disorders. And we track these patients in 2 different dose-finding cohorts for CLN3 in a low and high dose. And I put this symbol here for the University of Rochester because without the formative work that the clinical team has done at the University of Rochester, we would not have the tools to run a clinical trial. So right around the time that I started graduate school, Fred Marshall, John Mink, Heather Adams, Eric Augustine all partnered together to come up with the Unified Batten Disease Rating Scale. Now Jennifer Vermillion leads that Batten Disease Center of Excellence here in the Department of Neurology, and they continue to refine this clinical rating scale that's used worldwide. So really, that although they weren't initially involved in this clinical trial, they were pivotal in training the clinicians in order to be able to do this and capture robust data. So there have been 4 patients treated so far. Because of sort of the state of biopharma, this has waxed and waned over the last few years, but is really getting reinvigorated. I put these slides together and sent them to the university several days ago. And so in that time, I'm going to tell you 2 things that have happened. So Alcyone Therapeutics had announced about 8 months ago that they were going to champion and move this program forward. Yesterday morning, Alcyone was actually acquired by Biogen for their device division. But with the plans that all the gene therapy programs of, including CLN3 that were at Alcyone, will now spin out into a new company called Neely Therapeutics. So they've already started manufacturing of more CLN3 virus, but just a sneak peek into some of the data from those particular patients. So the Batten Disease Center of Excellence here at the University of Rochester for many years has been running a natural history study that's absolutely imperative. No rare disease parent wants to enroll their child in a clinical trial if they're going to receive a placebo control. These are fatal pediatric diseases, and they need to get these medicines as early as possible in order to have a chance of living. And so this is just the cumulative data from those 4 patients over the course of 3 years, 36 months. You'll see that essentially this gene therapy has, in that first 3 months, really helped to stabilize the progression of this disease. I'll actually draw your attention to these lower panels that we've pulled out, one of the low dose and one of the high dose patients, patient number 2 and patient number 4. And the WISC is actually an intellectual disability tool that's used in pediatric patients to track things like vocabulary, working memory, digit recall. And you'll see that over the course of 60 months, those 2 patients following gene therapy have essentially stabilized. For me, one of the remarkable things too is that you may, on the early slide, the patients actually had some vision loss. And in fact, several of them were almost entirely blind by the time that they actually entered the study. But you'll see that the gene therapy, even though we're delivering it into the brain, it does not go directly to the eye. We're able to prevent some of that vision loss. So I'll tell you that as late as Easter of this year, I've seen videos of the kiddos in this trial. The girl that was treated earliest, the patient number 2 at the age of 6, she is now 12. She was Easter egg hunting. She was walking. She was able to see the Easter eggs and pick them up. And for me, this is absolutely remarkable that these kids should at that age be in a wheelchair, nonverbal, not able to see, and having severe seizures. So it's just an absolutely remarkable program. Switching gears slightly for genetic medicines in CLN3, I mentioned to you that we also work on exon skipping ASOs in partnership with our colleague Michelle Hastings at the University of Michigan. So for years, we've been working on antisense oligo that essentially can skip out a common deletion that happens in CLN3. So in about 75 to 80% of CLN3 patients, they have a genetic mutation that deletes exons 7 and 8. So Michelle and I have been working to test an ASO that would essentially skip over exon 5 and then the piece that's missing for exon 7 and 8, and then put the entire rest of the gene back in frame. And so the patients would express a version of CLN3 that is almost intact. So we worked with Michelle to biologically show that this truncated version of CLN3 is still efficacious, that biologically it's able to do everything that we need to do. And we're slowly working our way towards the clinic to be able to open a trial with this ASO. But in the meantime, we took advantage of a unique legislation, a unique guidance that's been put out by the FDA, really pioneered by Tim Yu at Harvard for a different form of Batten disease. And these are called N of 1 trials. And so these are really, the concept is that you would have a rare pediatric disease where you could actually use precision medicine like ASOs to develop a custom medicine just for that patient. And so they give you expedited review, processing for your IND to be able to take these to the clinic. So we have a family that we work with in North Carolina who have 2 children that have 2 different mutations. They have the common mutation, but they also have this ultra rare mutation at residue at position 569. And so what Michelle's team was able to do is design an antisense oligo that essentially skips over that splice site between exon 7 and 8, puts the gene back in frame, and essentially makes an almost full-length CLN3. And so about a year and a half ago, in partnership with the University of North Carolina, we were able to take this amazing therapy into the clinic for these 2 twin sisters with CLN3 Batten disease. These girls, since they were young, have been obsessed with zebras and monkeys. Normally, there's an international naming board for drugs, but with these ASOs, they allow it really to the purview of the developer and the family to name them. So the parents requested that the drug be named Zerbroncocin, which is a combination of the words zebra and monkey for Mac and Amelia's favorite animals. These girls were fairly along in their disease progression by the time that they were treated. So I think the family went into this hoping that what they were doing was the legacy of their children was to provide a playbook for others to be able to develop custom medicines for their children. I don't think they ever anticipated the positive outcomes that they're actually seeing in these children. So last fall, for the first time in 3 years, they were able to go out to dinner with their children. Their daughter Amelia, who had been unable to actually eat any solid food for several years, is now eating enough that she can return to her favorite consumption of ice cream, that she has enough mouth control and motor control to be able to do that. And it's just absolutely remarkable to see what these can do. So as I mentioned, one of the things that involvement in the clinical trials has really led me to see is that we as basic scientists can still help to drive forward the advancement of science and the clinical trial and really help to expedite these. So on the top, I'm showing you guys what many of you are probably familiar with is the normal drug development process, where you go through sort of a phase 1 safety and dose finding trial that can take several years. Phase 2, where you're actually looking at efficacy, and then move into phase 3, your pivotal and expansion trial, before you would actually get approval of these drugs. So if you think about the traditional development process for this, this can take between 5 and 10 years to get a drug across the finish line. And these Batten's kids do not have the luxury of time. And so in rare diseases, what the FDA and the federal government have allowed is a number of concessions to allow for, if you can provide efficacy off of your phase 2, you can actually move straight into market approval and never have to do a phase 3 program. But one of the sort of FDA mandates that we've really grabbed a hold of is this idea that if you had a surrogate marker for your disease, a biomarker that you could actually utilize to track the disease, that you could actually move straight from that phase 1, phase 2 into a full approval of your drug and get it to patients in time to potentially save their lives. So we've been doing a tremendous amount of work on biomarkers. Many of you have probably heard of a biomarker, and I think for decades, scientists and clinicians have thought of biomarkers as finding that needle in a haystack. But we live in the day of AI and machine learning approaches. And so the strategy that my team has taken is to set out and look at the whole haystack. How do we take all of that data, the EEG data that John Fox's team is collecting, the clinical rating scale data that Jennifer Vermillion's team here are collecting, ERG data, and combine that with biosignatures that we might pull out of biofluids like blood or CSF samples. And so I'm going to tell you just high level a little bit about this work. But before I do that, I have to introduce my friend here, the pig. I mentioned to you that we've made a number of translational tools for studying CLN3 Batten disease. I mentioned to you that those CLN3 mice are not the best model for studying the disease. And so what we did in partnership with a company in Iowa called Exemplar Genetics is develop a number of rare disease pig models, but particularly of use is this CLN3 pig model that I'm showing you here. And so from these pigs, we draw blood samples, and we're going to talk about some of the metabolomics work that we've been doing and lipidomics work, but we're also doing a tremendous amount of proteomics work to try and identify a class of molecules that we can track over time and with the progression of the disease, but also that inflect with treatment. We also continue to collect data, and I'll use the gait analysis as one example of how we do this. But in partnership with Ruchira Singh here at the University of Rochester, we've also been studying visual deficits in CLN3 and using this pig model, which actually has vision deficits, unlike the mouse, to be able to dissect exactly how the vision loss is occurring in CLN3 Batten disease patients. So we used gene editing tools to, again, go in and introduce that common deletion of exons 7 and 8 into these pigs. And then essentially, just like a human patient natural history study, we track those pigs over time, monitoring them over the course of 4 years. These pigs they're called Yucatan miniatures. So they're a mini pig, a normal, if anybody's ever been around a normal swine farm, pigs go up to about 400 to 500 pounds. These guys are about the size of an average adult male. So they get up to about 180 pounds. But we do a lot of behavioral assessment, trying to get a pig to walk on a balance beam or a kinematic gait platform is not easy. So really this has been heroic efforts by my team at Stanford to be able to do ERGs, EEGs, gait analysis, and then use those biofluids to be able to track patients or the pigs over time. So just to give you an idea of what this data looks like, when I say that we're using sort of looking at the whole haystack and using machine learning to pick out signatures within these data sets, each of the different types of things we're looking at, whether it's metabolomics, EEGs, or gait, I will call these modalities. And within each modality, we can have hundreds of different parameters or readouts that we're looking at, or in the metabolomics or lipidomics, thousands of different readouts. So we use, in this case, for the gait analysis, we use principal component analysis to essentially unbiasedly go in and pick out different variables, how they change. So this is sort of a schematic as these pigs walk across a xenomat. This is the same kinematic gait analysis system that the U of R's team uses for monitoring gait in the human patients. And it spits out 185 variables from every single foot strike. And then we can use PCA. So rather than go in and say, hey, do we see a difference in stride length or stride width? We let the machine actually tell us what the most important variables are. And we're able to discern differences between the CLN3 pigs and also even discern differences in males and females, which have been reported in a number of Batten disease patients in how they actually walk or how they perform and progress in their disease. And so switching in partnership with my graduate school colleague and postdoc friend, Patty Sheridan, when she was at Metabolon, we launched into looking at metabolic signatures. And the cool thing is we were able to identify these glycerolphosphoryl molecules that were changed in our pig samples. So these are actually molecules that essentially allow for GPI anchors to be attached to proteins on the membrane. And when you have a mutation in CLN3, it actually pushes these away from the membrane. They don't degrade properly, and that head group starts to accumulate. So you can monitor that head group in samples. Right at the time that we published this work, there was a team at Stanford that also identified these as related to directly to the biology of CLN3. So we really think that this is something that is very closely related to the function of CLN3. These track over time. So if we look longitudinally in the pig samples, you can see increases, some that come up initially and go down. And when you think about machine learning approaches, you don't want everything to change the same. You want to be able to say, at a snapshot in time, I see this molecule high, I see this molecule is low, I see this particular phenotype in the patient, and be able to feed that in to create what we call a biosignature for that patient that tells me exactly, map to that natural history study, where they fall in their disease progression. And then if I give them a therapy, how does that actually change over time? And probably the most exciting part is down here in the lower right-hand corner, in partnership with An-Dang Do and Denny Porter's lab at the NIH, we've been able to recapitulate this in human patient samples. So where are we going next with this? I told you that there were two exciting things that have happened since I turned these slides in. In about a year ago, in partnership with alumni from the University of Rochester, Erica Augustine, who is now at Kennedy Krager, and with the Batten Disease Center of Excellence, we applied for a rare disease clinical research network grant that includes two projects. One where the U of R team continues to collect data, natural history data on CLN3, but expands that to 3 other forms of Batten disease. And our team comes in and collects the blood samples from those patients to be able to come up with biosignatures so that we can ultimately come up with the tools with the natural history study and these biosignatures to help us accelerate those clinical trials and get these medicines to these kiddos much faster than normally would be done. I just want to leave you with, at Stanford, we actually, where my academic lab is, we're really invested in rare diseases. So we don't stop at just the bench science. We've actually continued to develop tools for rare diseases, including this program called CORDS, which is the Coordination of Rare Diseases at Stanford, which is a free-to-patient, free-to-advocacy team registry program that currently includes over 3,000 rare diseases and 125 advocacy partners to essentially collect data on patient-reported outcomes. which is becoming more and more instrumental in FDA clearance and FDA approval of drugs. They want to hear that the patients genuinely believe that the therapy is beneficial, not just that the clinical metrics are showing a benefit. And so I go back to the story I told you of the Salin 3 kiddos, twin sisters in North Carolina. By clinical metrics, they probably would not show efficacy of those ASOs, but just the benefit that the family has seen from those therapies alone would be captured in something like a patient registry. So I will stop there. I mentioned to you that I don't have time to talk about a lot of the work, but I'm just going to leave you with this picture because it really takes an army. This is my absolutely amazing team at Sanford Research. As I mentioned throughout, this is in partnership with a number of rare disease foundations, therapeutic companies, and really a lot of this work got its start at the University of Rochester, and I'm so fortunate to be able to continue to work with the team here on advancing our Salin 3 work. So I will stop there. Yep, and if you guys have questions, we have microphone runners in the room that can come to you with microphones. There's one over here. My question is, and this is being recorded too, so I know they want everyone to know.
Thank you very much. Very interesting presentation. I'm not a basic science person. I'm just an internist. And I actually understood what you were talking about.
That's a win.
So thank you. Trying to make this complicated information usable and understandable is definitely an art. So thank you. I'm thinking of the bigger picture in terms of prenatal genetic testing and whether or not any of this testing for Batten disease is available or is even being done, or do you need to be part of a research?
Yep. So many of you that work in pediatrics know that generally until there is an approved therapy for a disease, a newborn screening assay does not exist. So Salin 2 Batten disease is the only form of Batten disease that has approved ERT. And that was approved almost a decade ago. And still to this day, not all states actually do newborn screening for Salin 2. If patients know that they're carriers, they can do pre-implantation genetic screening. That does occur. Unfortunately, for families like the Khans, whose twin daughters are receiving the ASOs, they were conceived through in vitro fertilization. The parents had absolutely no clue that they were carriers for Salin 3. And so, unfortunately, until we can change legislation that allows for earlier diagnosis of these diseases, and as you guys saw from that Salin 3 patient, the earlier we can get these genetic medicines to these patients, the better the outcomes are going to be. So it's really a chicken and egg situation that we can't be able to do that. But with Denny Porter's team at the NIH, we have also started to explore whether those biomarkers that initially we intend for use in clinical trials and tracking longitudinally may also be able to be used for the newborn screening assays as well. So when we collect blood samples from the patients, we actually do bank heel prick cards as well to be able to go back and see if these could be used for newborn screening. John?
Hi, that was great, Jill. Question about your analysis of pigs and any animal models that you are using. So as you are aware, right, like AI is being used to diagnose and also for treatments. And so for mice in my lab, we're getting into deep lab cut to figure out movements and very small changes that my eyes would never pick up. Are you or other people in this arena using now AI to determine if treatments are effective?
So in terms of the treatments, we still rely on probably the more antiquated behavioral testing tools, but we have started to use video capture with the mice and with the pigs to be able to actually really remove the human bias component of the analyses. So as a part of that rare disease clinical research network, there's a team at Hopkins that essentially takes the kinematic gait analysis data that the Rochester team is capturing from natural history studies. They're able to create a registry key that then other clinical sites can just record a video on their cell phone of the patient walking. They feed that data into the registry key and it spits out data as if they were walking on one of these, either a three-dimensional bodysuit or a kinematic mat. So that's how we're actually utilizing it going forward, is to essentially remove that human component and human bias from the data analysis. There's one right there.
Thank you for that terrific talk. How is this already very challenging work proceeding in the current environment of support? You got NIH right there in the middle of
Yeah.
your slide. What impact has it already had? And to what extent will this go forward privately? Can it possibly go forward?
So that's where I think, like, even as basic scientists, we need to think outside the box for our funding opportunities. I include in the lower right-hand corner 2 foundations that have been absolutely instrumental. There's a third one I didn't include on the slide that actually was the entire funder for the CLN6 gene therapy program. They funded all the GMP production of the virus. They funded the clinical trial. So these private foundations are becoming extremely important and vital to fueling this. I would say pharma is also vital to fueling this, but the biopharma sector has probably taken even a worse hit than NIH over the last couple of years. So they've started, you've seen a number of rare disease programs essentially just shuttered and put on a shelf over the last few years. So the Rare Disease Clinical Research Network grant that we just got this week is an actual NIH grant. So the NIH does still fund basic science. It's just getting harder and harder to be able to get those grants. And so thinking about things like I mentioned with our small molecules, we've actually stood up a small biotech company that allows us to start looking for funding like SBIRs that we never would have thought. So small business innovation grants to be able to fund and move that work forward. Partnering with pharma companies to be able to do some of this work in an academic setting is another way that we've kind of thought outside the box to be able to fund it. But it is exceedingly hard. So one last question up here.
Just to piggyback on that comment, I'm a neurologist, and we sometimes get patients with spinal muscular atrophy, SMA, which is a genetic pediatric hereditary condition where there are now approved small molecules, ASO, and gene therapies, which are exceedingly expensive and really hard to get approved and to get these to the patients. And so with this explosion of new genetic potential therapies for various rare diseases, I worry about the burden for all of us to be able to get access to these patients. And, with the pharmaceutical companies needing to get their R&D reimbursements, how high and how expensive it's going to be for these patients.
Yeah. And I should mention that the CLN6 program that we took in lightning speed to the clinic was actually off the back of Zolgensma, which is the AAV-based gene therapy for SMA that was developed by Brian Kaspar's team when he was at Nationwide. Brian and I were actually the ones that co-developed that CLN6 program. So we were really able to use a lot of the learnings from SMA to be able to move these gene therapies forward. To that point, one of the probably most interesting things to me over the last few years working in gene therapy is the economics of healthcare delivery and insurance payer-provider relationships. And so you think about somebody who maybe needs to go on dialysis, have a kidney transplant, and you look at the economics that insurance companies are willing to pay over a period of 2 to 3 years to be able to treat that patient who's in renal failure. That can add up to several $1 million that insurance, that provider is actually covering for that patient. And rarely do you hear people balk at those costs. Gene therapies, because of the cost of manufacturing and the clinical trials, they're being price tagged at between $1.7 and $2.5 million per dose. But it's a one-time treatment that could save that kid's life. And with SMA, what you're seeing is these kids, it's also referred to as floppy baby syndrome, who normally don't live past 18 months to 2 years of age, are now going to kindergarten. They're walking, they're still talking. So to be able to give a kid a chance at life is absolutely remarkable. And so I think we just need to change how we think about insurance reimbursement for some of these genetic medicines. Some of the insurance companies have kind of worked to say that they're going to look at efficacy. So as long as the therapy continues to show efficacy, they will provide reimbursement. But it's really in the United States, we've been able to achieve this, but many countries actually, even ERTs, Canada, a forward-thinking country, just recently removed access for a family of CLN2 family that was receiving already on therapy for ERT, and essentially said that we're no longer going to reimburse for this ERT. The family and the advocacy groups had to fight tooth and nail to be able to get that medicine, because essentially it would have been a death sentence for their child if they weren't able to do that. So for any of you guys that can advocate for sort of healthcare economics and the insurance side of this, it's going to become more and more important as we move genetic medicines forward. So I think that we will end there. And I thank you guys very much. And we'll be back in here, and I'll turn it over to Ernest to tell you about our next one.